In today's video, we are going to talk about hepatitis B virus serology or hepatitis B virus serological markers. But before going to the actual topic, first of all, we should know about hepatitis B virus structure. It consists of an outer lipid envelope. This envelope contains a protein called the surface antigen, which is important for laboratory diagnosis and immunization. Inside the envelope is an icosahedral nucleocapsid, also called the core particle, composed of hepatitis B core antigen. This capsid surrounds the viral DNA and associated proteins. Viral genome is a partially double-stranded circular DNA. Virus carries its own DNA polymerase enzyme, which has reverse transcriptase activity. There is another antigen located in the core known as hepatitis B E antigen. This antigen is an important indicator of transmissibility. Body produces antibodies in response to antigens like hepatitis B surface antibody, hepatitis B core antibodies, and hepatitis B E antibodies. Some important points to remember. Antigen and antibodies can never be detected at the same time. When antigen is positive, corresponding antibodies would be negative. Hepatitis B core antigen is produced in so minute amount that it cannot be detected. Hence, we detect hepatitis B core antibodies. Hepatitis B core antibodies are either IgG or IgM depending upon the phase of infection. Hepatitis B E antigen is positive only when there is high risk of hepatitis B virus infection transmission. There is a period when hepatitis B surface antigen has disappeared from plasma, but hepatitis B surface antibodies is not yet detectable. This period is known as window period. Now let's talk about hepatitis B virus serological markers in different scenarios. In acute phase after being exposed to hepatitis B virus, person will have an active infection. So hepatitis B surface antigen would be positive. Its corresponding antibodies would be negative. As virus is in its replicating stage, so hepatitis B E antigen would also be positive, and hepatitis B E antibodies negative. Hepatitis B core antibodies will be mainly IgM, which appears first after infection, and PCR would also be positive. In window period, both hepatitis B surface antigens and hepatitis B surface antibodies would be negative. Hepatitis B E antigen would be positive or negative depending upon active viral replication and infectivity. During this period, hepatitis B core antibodies, specifically IgM, can be positive and is used to detect recent infection during this period. In chronic hepatitis B virus infection, indicating infective stage, hepatitis B surface antigen would be positive. Corresponding hepatitis B surface antibodies would be negative. Hepatitis B E antigen positive, hepatitis B E antibodies negative. But now the hepatitis B C antibodies means hepatitis B core antibodies would be mainly IgG along with positive PCR test. In recovery stage, as now there is no infection, so hepatitis B surface antigen negative, hepatitis B surface antibodies positive, hepatitis B E antigen negative along with corresponding hepatitis B antibodies positive, indicating low likelihood of transmission. Anti-HbC would be IgG with negative PCR. In case of vaccination provided against hepatitis B virus produced using recombinant DNA technology, genes encoding the hepatitis B surface antigen is isolated and used. That triggers the immune response. Hence, mainly hepatitis B surface antibodies would be positive. Hepatitis B surface antigen negative plus all rest of the serological markers would be negative. Hepatitis B core antibodies would also be negative. This is the main difference between the recovered and the immunized state.